Hello students, welcome to HelloBiologyAssam. Today we will see about anatomy of flowering plants of class 11. So let's start the video. Some important points to remember about anatomy of flowering plants. This chapter introduces the internal structure and functional organization of higher plants. Study of internal structure of plants is called anatomy. Plants have cells as the basic unit. Cells are organized into tissue and the tissues are organized into organs. The tissue system. Plant tissues which perform same function irrespective of their position in the body constitute tissue system. On the basis of the structure and location, three types of tissue system have been recognized within the plant body. Epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. Epidermal tissue system. This system forms the outermost covering of the whole plant body. Comprises epidermal cells, stomata, and the epidermal appendages, the trichomes and hairs. Epidermis is the outermost layer of the plant body and single layer made up of elongated, compactly arranged cell. Epidermal cells are parenchymatous. Thick waxy layer of the epidermis is called cuticle, prevents the loss of water. In the epidermis of leaves, stomata are present. It regulates the process of transpiration and gaseous exchange. Stomata are composed of two bean-shaped cells called guard cells which enclose stomatal pore. Guard cells in grasses are dumbbell-shaped. The guard cells possess chloroplast and regulate the opening and closing of stomata. When guard cells are specialized in their shape and size called subsidiary cells. The stomatal apparatus are guard cells and the surrounding subsidiary cells are called stomatal apparatus. The cells of epidermal bears hairs. In case of stem, it is called trichomes. The guard or fundamental tissue system. Except epidermis and vascular bundles constitute the ground tissue. Consists simple tissues like parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are present in cortex, pericycle, pit, and medullary rays in the primary stems and roots. In leaves, ground tissue consists of thin-walled chloroplast containing cells called mesophyll. The vascular or conducting tissue system. This type of system consists of complex tissues, the phloem and the xylem. Both xylem and phloem constitute vascular bundles. Secondary xylem and phloem are found in dicotyledonous stem due to presence of cambium and are called open vascular bundles. In monocotyledons, secondary tissues are not formed due to absence of cambium and are called closed vascular bundle. Vascular bundles are arranged in an alternate manner along different radii. The arrangement is called radial. In conjoint type, the xylem and phloem are jointly situated along the same radius of vascular bundles. In the picture, you can see phloem, cambium, xylem. These are the types of vascular bundles: open vascular bundle, closed vascular bundle, and radial arrangement. Anatomy of dicotyledonous root. Transverse section of dicotyledonous root shows the following characteristics. The outermost layer is epiblema and in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. Innermost layer of the cortex is called endodermis and is single layer of barrel-shaped cells without intercellular spaces. Kasparian strips are found in the tangential as well as radial walls of the endodermal cells. Next to endodermis, pericycle cells are found and it is thick-walled parenchymatous. The feet is small or inconspicuous. The parenchyma cells lie between the xylem and phloem and are called conjunctive tissue. Tissues on the inner side of the endodermis like pericycle, vascular bundles and pit constitute the stele. Anatomy of monocotyledonous root. Anatomy of monocot root is similar to dicot root. Consists epidermis, cortex, endodermis, pericycle, vascular bundles and pit. Xylem bundles. In comparison to dicot root, monocot root has 6. Monocot root has 6. It is called polyar. Pit is large and well developed. Anatomy of dicotyledonous stem. Transverse section of young dicotyledonous stem shows the following characters. Epidermis is the outermost protective layer of the stem covered with thin layer of cuticle. Also bear trichome and stomata. Consists with 3 subzones. Just below the epidermis, hypodermis is found, which is consist of collenchymatous cells. Below hypodermis, cortical layers are found and consist of parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called endodermis, which are rich in starch grain, and the layer is referred as starch reed. Pericycle is present on the inner side of the endodermis and above the phloem. Large number of vascular bundles are arranged in a ring, and it is characteristics of dicot stem. Vascular bundles are conjoined, open and under protoxylem. Parenchymatous cells with intercellular spaces which occupy the central portion of the stem constitute the pit. Anatomy of monocotyledonous stem. It has sclerenchymatous hypodermis, scattered vascular bundles surrounded by sclerenchymatous bundle sheath and conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Phloem parenchyma is absent. Water containing cavities are present within the vascular bundles. Anatomy of dorsiventral leaf. Vertical section of dorsiventral leaf shows 3 main parts. One is epidermis, one is mesophyll and vascular system. Epidermis covers both the upper surface, that is adaxial epidermis, and the lower surface, that is abaxial epidermis, have a conspicuous cuticle. Tissue between upper and lower epidermis is called mesophyll, which possesses chloroplast and carry out photosynthesis. 2 types of cells found: pellicate parenchyma and spongy parenchyma. Vascular system includes vascular bundles which can be seen in the veins and midrib. Vascular bundles are surrounded by a thick wall layer called bundle sheath cell. Anatomy of isobilateral leaf. Anatomy of isobilateral leaf is similar to dorsiventral leaf but shows some different characters also seen. In isobilateral leaf, stomata are present both the surface of epidermis. Mesophyll is not differentiated into pellicate and spongy parenchyma. In grasses, adaxial epidermal cells modify into large empty colorless cells. These are called balliform cells. So students, this is all about anatomy of flowering plants. Hope this video will help you to understand and memorize about flowering plants. Please pass this video among your friends, classmates and subscribe the channel. Thank you.